So what you're going to use is special slides, pen slides, polyethylene naphthalate slides. You can get them from Leica, the manufacturer. And so these are different than regular microscope slides. What you'll notice is there's actually a coating, a membrane on there, and it's only adhered along the edges. In the middle, it's actually not glued. It's just kind of firmly stretched over it. And we're going to prepare our sample on there. I just want to make sure I've got the right side. It's the slide that's frosted. And I like to use these simple reinforcement labels to kind of mark where I'm going to put my slides. And what's important is if you use something like this, as soon as I put it down, don't manipulate it. Don't try to tug at it or replace it, because you don't want to tug at the membrane. So I just kind of like gently lay it down. And you don't have to squash it down yet. Just position it. Try to get 3 on there if you can.
So this is where you're going to put your sample?
Yeah. And notice it depends on the slide. These ones maybe are a little smaller. We'll try to get a 3rd in there. I like 3. And it doesn't matter. It's just the hole in the middle that needs to be on that open membrane.
So it needs to be on the because I see there's an inner rectangle.
Yeah, there's an inner rectangle. You want those circles to be in there. And so I don't really have them totally adhered to there, and you can kind of put it on a clean chem wipe to do that. So there we go. And of course, you'd want to label it what sample you're going to have on there. And then you will spot your sample. We spot 3 microliters of your cellular pellet. I mix this. You want to just make sure your pellet's mixed up. You'd normally have vortex it or something like that. Whatever your procedure calls for. And you can, what I do is you don't want to poke the membrane. You don't want to do that at all. So first you just want to place the spot on there. And it kind of nicely spreads out. But if you feel you need to get, these are spreading very nicely actually. Feel like you need to get a better smear and that you need to smear it around. Make sure you don't use just the tip. Make sure you use the side and just kind of spread it out. And this actually does. And the membrane kind of has a way of doing that itself. So you, I just like to air dry. A couple hours would be nice. Some people like to put it in an oven. Since we're actually doing DNA analysis on this, I recommend you just air dry it. And then after that, when it's dried, you can do whatever staining procedure you'd like. Now, I recommend doing something like nuclear fast red. We found that that's effective, and that's what we've shown on the screen too, the nuclear fast red staining. Just be aware that if you do use Christmas tree stain, which is very commonly used, it does have a downstream effect on your analysis. So just kind of keep that in mind when you're actually what stain you're going to use.
Okay.
And another thing is that another reason I put the circles on there is that it's nice to have the circles to hold your stain. Kind of sits on top of there, so you don't have to immerse the entire slide. All right, okay. The first thing that you want to do is load your slide. So up here, you want to unload the slide tray. And you want to make sure you also kind of wait till it goes through each of its processes before moving on. So in this slide tray, you actually have 3 positions that you can load it. And what you want to make sure actually is your sample is face down. So in this case, we've got our little circles here that have the labels on it, and our samples are in the middle. And the frosted slide is here. I actually want to load it at the frosted slide at the bottom there. Load a couple slides.
Do you always want to set the slide holder down like that?
You know, this is actually something that Andy just told me recently. I used to only have a slide holder that had 1 holder, so I didn't really have an issue. But one thing, you don't want to have it over here, otherwise you might drop your slide. But you can also, if you're concerned about contamination, holding it down, you can do it by holding it up. Otherwise, you probably would want to make sure that it's actually a little more difficult because you have to get the spring back. You would want to make sure that you'd clean your work area or your chem wipe or whatever you have here if you're going to lay it down every time. go. So we'll just let it load 2 slides. It's actually a guide that you can just use and along the sides and push that in. Go.
Okay.
And then hit continue. Load back in. The next part what we're going to do is actually load the tube. See the tube tray comes out. And this is for the 0.2 tube holders. We use these small PCR tubes. And these things, you have to kind of make sure you have them secure. So you put that top in there and you can kind of push it in the edge there. And then the tube actually goes down behind. And in this case, you can even ensure that it's secure by, after you've got it in there, kind of pushing it further in.
Oh, that's good.
Kind of a technique. So you can see the tube is held down there on the bottom.
Now, does it matter if the cap is a little crooked, or is it okay if it's...
Well, you know, I think not being crooked is probably a better idea. I haven't seen a problem, but you want it as straight as possible. Now I'm going to put, I prefer actually only collecting 1 tube at a time, and then, you know, just to prevent any contamination issues. But I always usually put an extra tube in there for kind of a waste area.
Okay.
So that if I have anything when I'm doing like calibrating the instrument, or I just, maybe I'm ablating something that I want to get rid of and I need a spot to put it in, I have a waste tube. And I tend to put it in there. You can also choose if you want, maybe have a blank in there nearby. Okay. So what you want to do is put some kind of buffer, and in this case, in our protocol, we're going to put 23 microliters of lysin go. This is our lysis buffer. And you want to make sure, this is critical. You can just go ahead and pipette it right in there. But the other thing you need to make sure is that it doesn't just sit in a bubble in the tube. You want the entire bottom covered with liquid.
Okay.
So to ensure that, you can kind of swish around your, swish around the edge to kind of break that tension.
How critical is the 23 microliter volume?
Well, it really depends on your protocol. The I think 20 microliters is the absolute minimum you should put in there.
Okay.
But it's a narrow. It's 20 to 25. If,
Okay.
if you put 30 in there, I think it would be too much.
Okay.
Because what happens is that you might overflow, or it might be difficult to unload the tube.
Right.
But you want enough to be able to cover that entire bottom.
So you don't put anything in your other tube?
I tend
As
not
the
to in the
waste?
waste tube.
Okay.
It's something that you might want to decide if you want to use it as a blank, you might want to do that. So I've loaded in there, and the number's actually on the right side here. Okay. And so this diagram down here kind of indicates which slide you're at. You're either at the one that's lit here is the one we're at, but there's no slide under there, so we're going to move over to the first one.
Okay.
These are stained with nuclear fast red. Okay. Put these a little bit to the side, get some more room. And it actually helps sometimes to kind of go to the edge. That's why I like those little labels. You can kind of focus on that. These pen slides are actually easier because since they have architecture in the membrane, a lot of times you can focus on that.
Let me ask you, do they make labels bigger than, if you were to make a bigger spot?
You know, these are just plain Avery office labels, the kinds you use to reinforce the 3-hole punch. So that's where I got these. You can I actually don't know.
Okay.
This is a slide. If you notice, there's some circles on it. We've used this before, this particular slide, and it's just kind of. I'm actually going to move up though to the next circle that hasn't been dissected yet.
So now I see you're using different.
Yeah, so this is kind of
controls
a unusual mouse. It's something you have to get used to instead of using a normal microscope adjustment. So the bottom here, you can go back and forth. The top is up and down, and the back is focus.
Okay.
So we are at 10x, and this is just kind of to get yourself oriented onto the slide, And I don't typically do sperm searches at this level. You really can't see the sperm. So we'll move down to, and you can adjust the objectives either through the screen here, which is a new feature, and I really like. Here's a 20x, and then we can go to 40x, which is usually the kind of the working. Did you notice that this seemed like that there's a lot of distortion in the membrane
Yeah, I
compared
wonder
to
if they changed something.
compared to what I'm used to?
Do you think they did something when they were doing the fluorescence?
I don't know. I'm going to actually move to another slide to see that if it's a slide issue. Let's go back up to 10. Orient ourselves. Oops.
I think it went the wrong way.
It went wrong way. 10. There we go. Let's see. I don't know if it was the slide. This slide looks better, doesn't it?
Yeah.
Yeah. I think there was some distortion in that particular membrane. It had been used previously already. Maybe. Let's give it a try. Let's go to 40. Okay. So this slide has some epithelial cells and some sperm cells. A couple sperm head here, epithelial cells. Just kind of look through this briefly. We have quite a few cells to work with. Okay. So I'm first going to demonstrate just collecting sperm heads that are just out here in the open. Those are quite easy. So there's some different options you can use. Here you can do line, so you can draw a line. I'll erase it. Oops. Let me erase this. Okay. So you use that line, and you can circle around the area that you want to dissect if you want. Or you can use a circle, which is my favorite for sperm heads. It's easy and quick. And those are really the two. And what you can also do is if you have several sperm that you think you're going to dissect, you can reuse that circle and just, boom, collect another sperm. There's no more on this field, but you can reuse your last shape. Okay. The other thing you want to do is decide which tube you're going to collect into. So in this case, I have it on the far left, and it's this one. You can even label them. So I'm going to label this. You want to punch into SP.
Sure.
Okay. So that's going to be the far right too, and it's lit up in green. And there's a lot of adjustments that have to be made in translator, but usually I like to just test right away. Let's see if it's going to cut. So we circled the sperm, and we're ready to cut. We start cut. I don't know if the laser's on. Oh, there we go. There we go. I had to focus when it did that. So that cut around, and the sperm dropped into the tube. So let's try a couple of these others. We also have to do, sometimes you can actually set it so it'll erase what you did previously, and that's how the software is set. If the circles are still there after you cut, you're going to have to, there's an erase mechanism down here that you can actually erase what you previously selected.
I see.
So again, I'm going to use the circle button, and we have sperm there, and I'll do the reuse. You can make another circle there. And it's going to go in the same tube because I haven't changed anything. Start cut. And it'll cut. There it goes. Drops down. Drops down. And I have to see if actually there may be a little static here. So we're going to kind of investigate that. I think that cut actually might be just hanging under the slide. Does it look like that?
Yeah.
Yeah. So a couple things we could try to do. Let's use this move and cut feature and kind of maybe we can kind of tease it. And this is a static problem that could occur, and I'm hoping that it went into the tube. It's occasionally, if it's too dry of an environment, it might have static that actually sticks onto the underside of the tube. We're going to actually look in the tube in a few minutes to look for our sperm. So let's just cut a few more samples before we look into the tube. So I go back to draw and cut, and draw and cut is the mode that you're mostly going to cut in.
What was the mode you just used?
That was move and cut, where you actually can just click down and hold, and you can actually move the laser and cut. So we'll cut these 2 sperm. And cut. We've got some static a little bit, I think. So there's what we call a hanging chad. It didn't quite cut the edge here. And I find that actually in the older instrument version, I would get about 10% hanging chad rate. This is pretty easy to solve. You can kind of, oh, there's static. What I'm going to do, I'm going to see if I can cut a fresh. Might be really close. Cut that out. Did you experience that kind of static on your tubes yesterday when you were practicing?
Yeah.
Okay. Because actually, I didn't. I had only one hanging chad when we were practicing
no, we
yesterday.
had quite a few actually.
Okay. I'm gonna cut a few more.
Very nice.
Yeah. See those, and that, especially with this fast instrument. Another thing I'm going to do, this is just strictly for demonstration purposes only. You wouldn't actually do this in real samples. To make it easier. What? The pen? It's not calibrated.
Oh.
Yeah.
Does it say we could use that? This?
Yeah, I don't know if the pen is calibrated. We'll see in a second. Let me, I'll just show you the line cut feature. And this is just again, for demonstration purposes, just so I can show you in the tube a very large structure. Let's start cut. And you can do this on any cell. You can cut the epithelial cells. We'll actually do
a big group or something.
Usually a star you can find in the tube pretty easily. Yep, there it goes. Let's count a couple epithelial cells too. Now again, you wouldn't collect it in the same tube, but for just this demonstration purposes, I'll show you. So we're going to use, let's try the circle first. And you really only need the nuclei, so you don't have to collect the whole tube. Do this one, and we'll do this one. And obviously you would collect it in a separate tube. So let's look at actually the collector. This is just the cap we're looking into. Very dark. Oop. There we go. Might boost the light up a little bit so we can see a little better. There we go. See what we can see in here. And the thing is, it's kind of a battle between focusing at the bottom of the tube and in your word. And I see actually a little problem here that we. Interesting. It's what? You know, actually, there's somehow. I can't go all the way over. See at the edge here what that is?
Yeah.
Yeah, I didn't remember how I said how important it is to get all the liquid on the bottom?
Yes.
I didn't do that. And so what it is that this is actually just plastic tube here. And what happens is that when you're trying to look into the tube, usually all your samples kind of basically like slid down and they're over here, so you can't see them.
Oh,
they're kind of in the shadow.
That's
what they tend to migrate to. So that's one issue in terms of the other issue.
Maybe you were looking too far down.
No, that's actually there's no liquid in that region. So that's something to be really careful of covering it. The reason why I don't know why this is, but I have found that I get a better yield when I make sure the liquid is all covered on there. And if you find that you're having this problem repeatedly, you may think that you might actually need to add 25 microliters perhaps of liquid.
I see.
The tubes that I've used, I wouldn't expect that a tube would be different in volume from maybe one manufacturer to another, but perhaps there is. Because in my case, in the tubes that I was using, 23 usually was plenty. But maybe these, for whatever reason, maybe they're a little different. Just something you might have to practice with.
Would it be a good idea to inspect the lid before you cut?
Yeah, that is a good idea. So this is probably one reason why I cannot see. So we'll probably throw another what I'm going to do is I'm going to unload. So we'll go back. We have to go back to specimen first. And I want to unload the tubes. Yeah, there's a little. So what am I going to do? And again, this is for demonstration only. You wouldn't do this normally in real samples. Can you hand me? Actually, I can just use this slicing now. I'm just going to add a little bit more water, so we don't have to load an entire tube. Just going to add a little bit more. But if you were in reality, if you were in a situation where you had already collected some samples, and then you realize you didn't have enough buffer water in there, you could, this is a way to kind of preserve your sample still. Could add a little more liquid. And if you were in a really dry environment and you were taking a long time to collect your sample, you may have some evaporation. I'm sorry? Yes. Okay, let's go back. So what we're going to do is I'm going to unload. Oops, I'm sorry. I unloaded the wrong thing. I'll have to go back. Okay. So for demonstration purposes, I'm going to just add some more liquid to this so we can continue on. But really, if you had a sample that you had been collecting and you found that you didn't have enough liquid in there, but you still wanted to preserve what you had collected, you could add more liquid.
Okay.
Whether it be probably water would be the best option.
Why would water be better?
Well, I'm just thinking if you didn't, depending, it would be on your own protocol, but if you're talking about evaporation, that would be mostly water, so you'd want to just add some water back. This is really only in a circumstance that you would be forced into it. Would not make it a habit. If you took a long time to collect your sample, or you walked away, or something happened that it was sitting open like that and there was some evaporation, that would really be, I would hope, the only case. Okay. Load that back in. All right. So let me cut another couple big things so we can just look into the tube. Just go down to kind of an area. So let's just collect a big ol' focus. I wouldn't normally use a star to collect sperm, but I want to be able to demonstrate that you can see a shape in there. A very odd star, I'd say. Okay, make sure it's in our sperm tube.
What did it just do? What was
Yeah,
that?
because I hadn't selected a tube initially, which I usually do, it was in the no cap position. It automatically knew that I didn't have a tube under there, and it said, which tube do you want it to be deposited into?
I see.
So I had to actually say the sperm cup. And the parameters on this, you're going to have to sit down when you have a protocol and your staining protocol and whatever slides you end up using, you're going to have to determine how intense the laser is going to be, how fast it is, just to do the cutting. You don't want to just turn up the laser all the way up. You want to just use the amount that you need,
I see.
and also the speed that you're comfortable with. So with
How do
this
you do
large,
that?
well, what you want to do is you'll kind of sit down with like some just test samples, and you'll play with the laser. And let me get the sample out and I'll show you. Okay. So you can see it's kind of hanging there. So again, we can use the move and cut and kind of tease it out. There we go.
Well, that didn't take much.
No. Let me go back to draw. So one of the things you can do to let's see, laser control setting. Oh, it's supposed to be on that screen, I think. Is that on? It is on. I'm not getting it. No.
I wonder if it's behind this software over here.
It was there yesterday. Okay, maybe we'll have to shoot that later. Let's go back into the collector. But basically, there's a whole bunch of functions. I don't want to actually go into the software in extreme detail. if you actually get this type of instrument and software, you'll probably have a lot of instruction on that. But in terms of what we were talking about, adjusting the laser, there's different settings that you can use in terms of speed and intensity of layer. And you basically kind of just play with the different levels and see, you know, for your tissue type, which would be sperm or epithelial cells most likely, how powerful a laser you need to cut through that. Going the wrong way. There we go.
Oh,
There we go. So
very nice.
that was that very odd star that I was cutting.
Yeah.
I'm looking around here. And I have noticed that, I don't know what happens. Remember how I already had cut some sperm that were in here?
Mm-hmm.
I have a feeling they're not probably floating anymore. They normally float on top, but because I added some liquid right on top, maybe they're
Farther down.
farther down at this point.
Is that why we can't see the star now?
Yeah, well, I'm kind of I'm focusing in and out of the different focal planes. There's some calibration that needs to be done on this, because my stage won't go all the way to the side. Okay, but you saw the star. Yeah, I'm kind of like looking through the tube, trying to find it. That would be, but considering how much, really, when you're using a microscope and trying to look through all that liquid, it's
Right.
pretty difficult.
Yeah.
So that's another reason why you really want to try to get that liquid the first time out all the way there, because most of your sample is floating around on the
I
top.
see.
Okay, so we've collected our sample, and now we need to, let's go back to the specimen. So you should always like go back to the specimen before you do the next step. Let us unload the tube. And this is a really important to be careful. And usually I would wear extra small, tight gloves if I wanted to do this right. So you don't want to flick your sample away. So you want very, very careful how you unload this. So I put a little pressure using one of my fingers behind there while I kind of release the bottom. There we go. And then you want to carefully slide it out, applying some back pressure. And then you can go ahead and flip it over like this.
So flip the bottom up to the top.
Yeah, the bottom, and you can flip up top. You want your sample, obviously, you want to keep vertical so that it's open to the air there. And then you can go ahead and just spin it in a little microfuge for a few seconds, and your sample will be at the bottom there. You should spin it. If you try to do this, this really won't be enough. You really need to do a spin, yeah. And then it's ready to go. In this instance, we're doing a license and go protocol, so it's still incubation. But whatever extraction protocol you decide to do, you can take this and run it through that. Okay, so the other tool, we showed you the line option and the circle option, also reuse. Just so you know, there's also this rectangle. I have actually never used it. I don't find it useful in dissecting cells.
I see.
And you can practice with it. But another thing is you can use this nifty little pen mouse, I guess you call it. You can actually just circle what you want if you're very comfortable with that.
So you have to click the line.
So yeah, you click the line, and you can go ahead and if I wanted to, like, okay, I want to collect all these cells here, you can do that. And it's actually easier than if you're trying to do a line on the mouse. It's hard.
A little harder to
Yeah,
guide.
if you're actually going to do the line, the pen is very nice. We'll just. Oops. So one thing I forgot to do is erase the initial little test, but this will show you that you can just use the line tool and do the same thing as you did before. Nice little feature. You could actually
Very
flip this entire monitor screen flat if you prefer. And that's about it.